Hello students, welcome to my channel Deep Learning Biology. Now we are going to understand two terms. What are glycoproteins and what are proteoglycans. To make you understand these terms, first I will make you understand what is a simple protein. Now you know very well, in a simple protein you are going to have only polypeptide chain, means here you are going to only have amino acid joined through peptide bonds. Okay, so here in a simple protein you are going to have only amino acid residue. Okay, you are not going to have any other thing, and such type of protein they are known as the simple protein. And you must be knowing the example of such protein are, this is ribonuclease A. Okay, this is a very simple protein, ribonuclease A, and this is another protein, that is chymotrypsin. Okay, these two proteins they are made up of only amino acid. Here amino acid have been joined together by peptide bonds, and as they do not have any other chemical entity, these are known as the simple protein. But if you see, there are proteins known as conjugated protein. Now what is a conjugated protein? A protein that function in interaction with other chemical groups. So that protein will be known as a conjugated protein. Now try to understand, a protein that function in interaction with other chemical group. Now these other chemical group, these are non-polypeptide in nature. They are not other proteins. These are non-polypeptide in nature. Yes, it may be anything. It may be a lipid, it may be a sugar, it may be a metal. Now try to understand. Let's suppose there is a protein. Okay, so this is the protein, and now if it is attached with another non-amino acid part, so this is non-amino acid part. Okay, and this is the amino acid part of the protein, where the amino acid have been joined together to form the protein. Okay, now as I have told you, the non-amino acid part, it may have a lipid, it may have a sugar, okay, and it may have a metal. Okay, so these can be found as the non-amino acid part. Another important thing you must remember, this non-amino acid part is also known as the prosthetic group. Okay, so here the protein is going to function in conjugation with the prosthetic group, and the prosthetic group is the non-amino acid part, which can be a metal, lipid, or the sugar. Okay, now try to understand this. This non-amino acid part, it is going to attach with the protein either by covalent interaction, there may be a covalent bond, or it might be attached to the protein by non-covalent interactions. Okay, so there can be both the possibility. It may be a covalent bond which is going to attach the prosthetic group, or it may be a non-covalent bond. Now as I have told you, the protein when it is attached to a non-amino acid part, okay, that we called as prosthetic group, is going to known as a conjugated protein. Now, as I have told you, this part, it may be a lipid, and here when it is lipid, the example is lipoproteins, okay, and within the lipoprotein, one of the best example is beta 1 lipoprotein, which are being found in the blood. Same way, it can be a sugar, okay. So here the best example is glycoproteins, and in case of glycoproteins, you must remember the example immunoglobulins. Okay. Next, I have told you that this part, it can be a metal also. Okay. Now, in that case, the example, it will be known as MetalloProteins. Okay. So there are various metal being found as in the prosthetic group. Yes, if it is iron, okay, then it is being found in a protein known as ferritin, okay. Then zinc is being found in the protein alcohol dehydrogenase, okay. Yes, calcium is being found in the case of protein calmodium, okay. Then you must remember molybdenum is being found in the protein, that is dinitrogenase enzyme, okay. So these are some of the example you must remember. Now, as I have told you, today class is about what are actually glycoproteins and proteoglycans. So let us try to discuss them. So what are glycoproteins? Try to see. These are made up of two words. Glyco means sugar. Proteins, you know, it is the proteins. Okay. Same way here also, proteoglycans. Here you also have proteins, and then you have the sugars. Okay. So let's try to understand these two terms. What are actually glycoproteins and what are proteoglycans? Now, in case of glycoprotein, here you can define them: a protein containing a carbohydrate group. So if you are going to have a protein and if some carbohydrates is being attached to it, then it is a glycoprotein. Here you must try to understand this, okay, that here within this protein, it may be that there may be many sugar residue may be attached. So many sugar molecules might be attached, and they might be attached covalently. So this is known as a glycoprotein. Now, in case of proteoglycan, if you define the proteoglycan, it is a hybrid macromolecule consisting of heteropolysaccharides polypeptide. Okay, so here you see it is saying that it is going to have two things. One is heteropolysaccharides and the protein part, that is the polypeptide. Now try to understand. You must have been confused. These two terms look similar. I just want to tell you, in case of glycoprotein, always remember, okay, the first part, it is less in amount. Same way in case of proteoglycan, the protein part, it is less. So here you can remember there is a protein, okay, and to this protein some sugar units, they are being attached. Okay, so that is a glycoprotein. The major part in a glycoprotein is your protein, while in case of proteoglycan you can see that the major part is glycans. Now we are going to have more discussion about the glycoprotein. As I have told you, a glycoprotein, it is going to have the major part is protein, and to it are attached the sugar molecules. Okay, and I have told you these sugar molecules which are attached, they are attached covalently. Now these glycoproteins are generally found on the outer surface of plasma membrane. Okay, yes, they may be found in the extracellular matrix. Yes, they may be found even inside the cell. So inside the cell they might be found in Golgi complex, they might be found in lysosomes, and they might be found in secretory granules. Okay, so these are some of the point you must remember. Now here I just want to tell you, these sugar units which are attached, they may be heterogeneous in nature, and this heterogeneous in nature will provide a very rich information. Okay, so you are going to have some information on the protein, and these information are coming through these sugar attached. Now I just want to tell you some other proteins, they can easily come and interact with these sugar, and you must know that those protein which are actually known as the carbohydrate binding protein, and they actually selectively bind these sugars, they are well known as lectins. Okay, so I have now introduced the term also, the lectins. Okay, now another important thing you must remember, that the sugar part which is attached to these proteins, it may be D-glucose, it may be D-galactose, it may be D-mannose, or it may be L-fucose. Yes, it may be N-acetyl glucosamine. Yes, it may be N-acetyl galactosamine. Okay, so there may be others, okay, but these sugars might be attached. Okay, and these sugars, they might be attached to the protein through the amino acid serine or threonine. Okay, now you know very well serine and threonine is going to have OH group. Okay, so these glycoproteins, they are known as O-glycosylated. So when the sugar is being attached through serine or threonine, while if the sugar is being attached through amide linkage of asparagine, okay, then those glycoprotein will be known as N-glycosylated. So there can be O-glycosylation or there can be N-glycosylation. The best example of the glycoproteins are immunoglobulins. Okay, yes, there are other proteins which are also the examples of glycoproteins. You can find many, but you must remember collagen. There is another protein, that is celluloplasmin. Then another protein, that is transferrin. These are example of the glycoproteins. Now we will have a discussion about the proteoglycans. Okay, so now you know very well that in case of proteoglycans, the proteo, it is the first part, it is going to be less. So your major part is glycans. So you can say proteins that are heavily glycosylated, they are known as proteoglycans. But I just want to make you understand that here the glycosylation, it is slightly different. The sugar which are attached, they are actually sulfated glycosaminoglycans. Okay, so here in case of proteoglycans, you will see that one or more sulfated glycosaminoglycans chains, they are covalently, okay, they are covalently joined to the protein. Okay, so that is known as a proteoglycans. I just want to tell you that in case of humans, you are going to have approximately 40 proteoglycans. Now here you see sulfated means it is going to have sulfur group. So generally the proteoglycans, they are having a negative charge. Okay, this point you must remember. So if you see in a proteoglycans, you are going to have a core protein. So you are going to have a core protein, and this core protein is being attached to the sugars which are known as glycosaminoglycans. Okay, so these sugar which are being attached, they are known as the glycosaminoglycans. Now let us try to understand what is glycosaminoglycans. These are actually heteropolysaccharides of two alternating unit. One is either N-acetyl glucosamine or N-acetyl galactosamine, okay, plus the other unit is uronic acid. Okay, so it is a heteropolysaccharide which is going to have two alternating unit. दो unit हैं. One unit can be N-acetyl glucosamine or it can be N-acetyl galactosamine. Out of these two, one might be present and another is going to be present the uronic acid. Now this uronic acid also give a negative charge. Okay, so I have told you the sulfate group, it is going to give the negative charge, and this uronic acid is also going to give the negative charge. So proteoglycans, they are going to have extensive negative charge. So you must remember that proteoglycans, they are going to have negative charge. One is due to the sulfated groups and other due to the presence of uronic acid, and that uronic acid is going to have COH group and that will impart the negative charge. Now I just want to tell you that these proteoglycans, they are going to generally found in connective tissue. Yes, they are also the major components of extracellular matrix. Okay, so this is about the proteoglycans. Thank you for watching this. Have a nice time.